Hi, everyone. Thanks for joining me today for episode 713 of People Behind the Science. I'm your host, Dr. Marie McNeely, and today I am joined by our guest, Dr. David Liu, to talk about life and science. And this episode is made possible with support from our sponsor, Innovative Research. Listeners, Innovative Research has provided scientists with quality lab products made in Novi, Michigan for more than 20 years. Their portfolio includes human and animal biologicals, assay and ELISA kits, antibodies, and more. Products can be purchased directly from their website by credit card or purchase order at www.innov-research.com. That's www.innov-research.com. They offer small quantities, bulk purchases, and custom orders. From human whole blood to specialized antibodies, Innovative Research has what you need to succeed in the lab. And today, our guest David is going to talk more about research in his lab, as well as his life outside the lab. So get ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am thrilled to be speaking with our guest scientist, Dr. David Liu. So, David, welcome to our show today. How are you?
Good. Thank you for having me.
Well, thank you so much for joining us, and we're excited to learn more about you and more about the amazing work that you're doing in the lab today. But first, let me give our listeners just a little bit of background information about you. So listeners, David is the Richard Merkin Professor and Director of the Merkin Institute of Transformative Technologies in Healthcare, Vice Chair of the Faculty at the Broad Institute of MIT and Harvard. He's the Thomas Dudley Cabot Professor of the Natural Sciences at Harvard University and a Howard Hughes Medical Institute, or HHMI, investigator. In addition, David is the founder or co-founder of several biotechnology and therapeutics companies, including Beam Therapeutics, Prime Medicine, Editas Medicine, Pairwise Plants, Exo Therapeutics, Chroma Medicine, and Residence Medicine. He completed his undergraduate education at Harvard College, majoring in chemistry. David was awarded his PhD in organic chemistry from UC Berkeley, and he joined the faculty at Harvard University immediately afterwards. He's been an HHMI investigator since 2005. And over the course of his career, David has received numerous awards and accolades, including being named the 2022 King Faisal Prize Laureate in Medicine, and he's received the Ronald Breslow Award for Biomimetic Chemistry, the American Chemical Society David Perlman Award, the ACS Chemical Biology Award, the ACS Pure Chemistry Award, the Arthur Cope Young Scholar Award, and many other prestigious awards, both for his teaching and his research. In 2016 and 2020, David was named one of the top 20 translational researchers in the world by Nature Biotechnology, and he was also named one of Nature's 10 researchers in 2022. In addition, David is an elected member of the U.S. National Academy of Sciences, the U.S. National Academy of Medicine, and the American Association for the Advancement of Science. And David, today we want to get to know you both as a scientist, of course, but also as a person. So can you start by telling us what you like to do when you're not doing science?
I confess, many hobbies.
Mm-hmm.
The hobbies are generally at the interface of science and art. I've done photography since I was a child. I turn wood vessels in my basement. I grow bonsai trees indoors. Poses interesting science challenges because you have to construct artificial light sources that can fulfill the photosynthetic needs to allow the trees to not just survive, but to thrive. I make a variety of homemade art projects, electronic art and otherwise. And I generally am passionate about ways to blend creativity and intellectual pursuits into something surprising and something beautiful.
I love it. Well, it sounds like you've got a lot of amazing hobbies. Do you find that these hobbies help maybe stimulate your creativity in science as well?
Definitely. Or I'd say the scientific research that we pursue is another form of expressing this passion for creativity and for beauty and for making things that are useful.
That is phenomenal. And I'd love to talk more about your science. You're doing really incredible work in the lab, and you've had some really exciting accomplishments come out of your lab. So when people ask that kind of basic question of what do you do for work, How do you describe all of the amazing things that you do to someone who's not in science or maybe not specifically knowledgeable about your field?
Probably the most general way I can summarize what our research lab has done for the past 24 years is that we integrate the components of biological evolution with chemistry to enable new types of therapeutics and to study biology. Now, that's a fairly high-level view of our lab, and people will often say, well, what do you mean by evolution? What do you mean by chemistry? And for me, chemistry's hallmark is really the ability to change the structure of a molecule to change its function in a way that hopefully you can anticipate. So not in a random way, but in a way that is at least partially designed. And our appreciation of evolution really reflects the awe that we have for the power of cycles of Darwinian selection, replication, and mutation. The same processes that, of course, gave rise to all of the incredible functions in living systems, but which we and others can now harness to evolve molecules with tailor-made properties.
That is fascinating. And I'm looking forward to getting into some of the details of the work you're doing into the lab as we go through our conversation today. But I love to start by dipping into motivation. I think as a scientist, it's really helpful to have bits of motivation, things that can keep you going when the times get tough. So, David, do you have a favorite motivational quote or a saying, whether it's about science specifically or kind of life in general?
I have many different motivations. I don't keep too many quotes in mind behind them, but one piece of advice that I frequently give when asked to give advice to younger students, younger scientists, is to follow your curiosity. That is, if you are motivated by pursuing mysteries, something I love doing, pursuing molecular mysteries, then following your curiosity will very likely result in your best work simply because it doesn't feel like work if you are pursuing a mystery that you feel very passionate about solving. I think we've all felt that page-turning experience of reading a mystery that has really captured our attention. Well, in many respects, doing science and even doing many of the hobbies that I described is like a real-life version of those detective stories where you are curious about how something works, or you're curious about whether or not it might be possible to connect a few concepts or maybe invent a new concept to enable something you think could be truly impactful. And then at that point, it just becomes a labor of love, a passion to pursue that mystery to its conclusion, whether it's positive or negative. But that pursuit can be incredibly satisfying, stimulating, energizing, and again, can put you in a sometimes precarious position where work doesn't feel like work. It feels like the kind of pursuit you would rather do than anything else in this world.
Well, that is amazing. And I'm curious, as you've been pursuing your own curiosity, have there been particular people who maybe had a big impact or role models that have really helped you get to where you are today?
Virtually every person I've interacted with in my career, both as a student and as an academic, has had a measurable and sometimes a profound influence in the way that I think about the world and the way that my career has unfolded. Maybe I'll start from the very beginning. My parents, of course, who always just, and I now realize how hard it would have been for them to do this, but they always resisted the temptation to kind of push me in any one direction. Even though my parents themselves were science-oriented, my mom was a physicist, my dad was an engineer, they actually let me explore the world and find what I was curious about myself. And some of my earliest childhood memories were of playing in our backyard, doing experiments, probably now would be considered somewhat unethical, on insects and on plants to try to understand how they worked, how living systems worked, to try to imagine how all of this came to be. And then, of course, as I went through my education, I was guided by many amazing teachers. High school science and English teachers in particular stood out. And then in college at Harvard, I was very fortunate to, as the result of a high school science fair award, I happened to get a ticket to attend the 1990 Nobel Prize ceremony in Stockholm, Sweden.
Oh, wow.
Which was an amazing opportunity, inspiring by itself just to be at the Nobel Prize ceremony. But as luck would have it, the 1990 Nobel Prize winner in chemistry was a Harvard faculty member, Professor E.J. Corey, a synthetic organic chemist, a chemist who has made remarkable contributions to developing the ways that we can put together molecules with tailor-made structures of our choosing, like molecular tinker toys, like molecular Legos. And to do that in real life with real molecules, which are, of course, far too small for you to be able to directly see in most cases, it required a level of creativity and brilliance and deep mechanistic understanding, intuition that I think many people, including myself, still find breathtaking. In any case, E.J. Corey's Nobel Prize ceremony, then known to me as Professor Corey, was an inspiration. And I remember asking him a couple of questions at his Nobel Prize ceremony lecture about some of the molecules he described. In fact, I remember one of the questions specifically, he was describing the synthesis of a small molecule called insect juvenile hormone. And if I recall correctly, one of the intermediates had multiple carbon-carbon double bonds in a long intermediate molecule that he needed to turn into the final product. And one of the steps was to create an epoxide, which is basically a chemical entity in which effectively you insert an oxygen atom into one of those two carbon-carbon bonds. And you end up with what looks like a molecular triangle in which one of the points of the triangle is that oxygen atom and the other 2 points are carbon atoms. So the remarkable thing about this reaction that he showed in the Nobel Prize ceremony lecture was not that he could convert a carbon-carbon double bond into an epoxide. I was starting to learn such reactions in class. But what was amazing to me was that he visualized that this molecule, even though it had multiple carbon-carbon double bonds, that only the one he wanted to turn into the epoxide would actually react that way. And so after his talk, I raised my hand and asked him, how did you know that just that carbon-carbon double bond that you wanted to turn into an epoxide would react that way and the other ones would not? And he explained to me in general terms how the shape of the molecule, as he imagined it folding, would only allow that one carbon-carbon double bond he needed to react to do so. And I was left with this sense of awe, as if you needed more evidence that somebody who was about to be awarded the Nobel Prize in Chemistry was an amazing scientist. But I was left with this sense of awe that somebody could achieve such a deep understanding of how molecules behave, that they could accurately predict what to me at the time as a freshman undergraduate seemed like a magically complicated process. Anyhow, when I got back to Harvard after the Nobel Prize ceremony, I found Professor Corey's office and I asked him if I could do research with him. And my organic chemistry teacher at the time, Professor Joe Grabowski, put in a good word, I later found out. And EJ, as I now know him, agreed. And I didn't at the time realize how special his agreeing to let me perform research in his lab was. But I started to get the hint when I realized upon joining the lab that I was the only undergraduate there at the time. And it was a lab of amazing graduate students and postdocs. And EJ and his graduate students and postdocs would then mentor me for much of the rest of my undergraduate career whenever I had time to dive into a research project, to watch somebody, to learn from them, to help out with a reaction, or I would do so. And I had many wonderful mentors in his lab, including Scott Virgil, who was an amazing organic chemist by himself, who'd go on to become a professor and then a lecturer at MIT and Caltech, respectively, I believe. And I remember as much as I loved learning from Scott, I also celebrated the times in which he started to go on the interview circuit and started to be absent from lab, because those were times in which I could actually run reactions by myself, purify the products, submit them for chemical analysis, and test whether I had learned how to make these molecules. And I remember a stretch where he was gone for a week or so, and I had advanced chemical synthesis through several reactions, purified the products, characterized them successfully. They were all correct. And to me, that was the only acceptable outcome. But after he got back, Scott gave me plenty of praise, and other people in the lab conveyed to me that that was unusual. They didn't expect that an undergraduate could do that. But that kind of reflected the excitement and the passion of being immersed in a research project where you didn't know what the outcome was going to be. You didn't know if you were going to be successful. Very different than following cookbook lab recipes in a class. And that was my first experience with research. And then I would go on to do a PhD with Professor Peter Schultz at Berkeley. And then around the third year of my PhD, I started to get calls. There really wasn't amazing email then. It was occasionally used, but it's not like it's used today. There's certainly no text messaging. So I started to get calls to the lab, correspondences to Peter Schultz, my PhD advisor, from departments who were asking if I would give a seminar. And also then as a graduate student, I was too naive to realize that what they were actually doing was sort of giving me an early interview for a potential assistant professorship. And I thought, oh, cool. Harvard wants me to give a seminar and Caltech and Berkeley want me to give a seminar.
Yeah, I'll talk about my cool research. Yeah.
So I did so, and I presented some of the work that I was doing in Peter Schultz's lab at the time. And of course, now I realize in retrospect how naive I was that they were effectively interviews for assistant professorships. In fact, I was so naive about the process, I hadn't even prepared the answer to what would now be considered the most important question of any junior faculty candidate, namely, what do you want to work on if you were a professor? So one of the few ideas that I did have as a thought out proposal was the one that you're required to submit at the end of your first year of graduate school at Berkeley, called your graduate research proposal. And for my first year graduate research proposal at Berkeley, I had proposed using DNA to orchestrate chemical synthesis in a way so that the resulting molecules were each attached to a small sequence of DNA that encoded their identity. And actually that templated their synthesis. And when I was asked during these interviews, these pseudo interviews, well, if you were a professor, what would you want to work on? That was really the major idea that I talked about. I had also had some interest in protein evolution that was emerging as part of what I was setting up in my PhD to achieve certain parts of certain projects. So when asked, I talked about two ideas, DNA encoded libraries and protein evolution. But at most of these places, I had not prepared any materials. I think it wasn't until maybe my third seminar that I realized I should probably make some slides. There was no PowerPoint then. So it's like printing on transparencies and putting them on an overhead projector. And that was really the start of thinking about what I wanted to pursue as an independent investigator. And then in 1999, so when I was a fifth year graduate student, I ended up graduating in May and then starting July 1st as an assistant professor in Harvard's chemistry department. And in retrospect, I had no idea what I was doing at that time. It was an incredibly exciting time in that suddenly I found myself in May a graduate student and in July a professor. I remember walking into my very first faculty meeting in early July and just sort of being in awe of all of these professors. Remember the last time I saw these people, I was an undergraduate. They were teaching me. They were winning the Nobel Prize.
Right.
And now I was walking into this dark wood paneled division room, as we call it, a faculty room that looks pretty much like a stereotype of a Harvard faculty room might imagine. There are portraits of esteemed faculty members on the wall. There's beautiful dark wood paneling everywhere. There's a large venerable wooden table and venerable Harvard logoed chairs everywhere. And I remember in this first faculty meeting calling everybody Professor Evans, Professor Kishi, Professor Corey, Professor Schreiber, all people who not only had taught me, but were kind of legends, continue to be legends in their respective fields. Literally some of the very best scientists, chemists in the world whose impact is felt every day. And so I referred to them, of course, as Professor Evans, Professor Corey, Professor Kishi. And then afterwards, I think it was EJ Corey pulled me aside and said, we think you should call us by our first names.
We're peers now. Yeah.
You're one of our colleagues. You should get used to that. So it was very kind.
Well, that is wonderful. I love to hear that your journey started with engaging in this kind of backyard science, and I know you've come a long way since then. So is there a particular project that you're working on right now, David, that you're perhaps the most excited about that you'd like to share with me and our listeners?
One area of our research that we've become somewhat known for is the development of methods to convert one DNA sequence in a living cell, in an animal and human patient indeed, to a different sequence of our choosing, something that is commonly called precision gene editing. And we've developed two ways to perform precision gene editing in living systems. The first way is called base editing, and the second way is called prime editing. Base editors and prime editors are not naturally occurring proteins. They are engineered, or in some cases evolved, in our laboratory. So they are evolved or engineered molecular machines that use components either taken from nature or created in our laboratory to directly modify the sequence of a stretch of DNA of your choosing in our vast genomes of 2 sets of 3 billion base pairs, to find a target sequence and change it specifically to a different sequence of your specification. And the reason that that's exciting is first for research. Virtually every kind of question in the life sciences can benefit from changing a DNA sequence in a specific way to a different sequence of your choosing, and then seeing how that change affects the resulting RNA, the resulting protein, the resulting biological pathway. So base editors and prime editors have become widely used research tools. And then second, there are about 7,000 known genetic diseases that collectively afflict hundreds of millions of people and, of course, their families as well. And these genetic diseases are caused by misspellings in our DNA, by mutations like a C that has become a T, by the loss of DNA letters or extra letters. And base editors and prime editors provide some of the first direct ways in living animals and in human patients to directly correct the misspellings in our DNA that cause genetic diseases. So base editors and prime editors are also being used by companies like Beam Therapeutics and Prime Medicine, respectively, companies I co-founded with some of my students and coworkers, to try to fix these mutations that cause genetic diseases so that we can treat diseases that previously really didn't have many treatment options.
Absolutely. And I know this work is already having tremendous impacts, as you alluded to, David. And to do amazing research in the lab, you have to have the right equipment and supplies. So we'd like to take a moment to thank our sponsor, Innovative Research, and to talk about their portfolio of ELISA kits, which offer high sensitivity, specificity, and can be used for a wide range of applications. Innovative Research ELISA kits are manufactured in their lab in Novi, Michigan, and are shipped directly to your location. They're a valuable tool for researchers and clinicians who need to detect and quantify specific biomolecules. Visit www.innov-research.com. That's www.innov-research.com to view their entire line of ELISA kits. And going back to our interview with David, of course, pioneering new technology in science is not easy. And we love to talk about challenges in science because there are just so many. So, David, do you have a particularly difficult challenge that you had to overcome in your career? Perhaps a major failure that you really struggled through? And can you tell us about it and how you worked through one of these difficult situations?
Sure. I mean, doing ambitious science by definition means you're going to fail a lot. And something that your listeners might be, especially the younger students and scientists, might be interested to learn is that, like a lot of young professors when I started out, I had a lot of uncertainty as to whether I was going to succeed, whether any of the projects that we set out to do were going to be successful. And that insecurity grew steadily over the first few years to the point that when I was a third year professor, I had convinced myself that I was likely going to fail. I remember going so far as to creating a PowerPoint slide deck, which I was going to present on why I thought I was going to fail to our chair at the time.
Oh, no.
So that I would explain both why I thought I wasn't going to succeed and how to best minimize embarrassment for the department and for the university. So like many people, I had some degree of imposter syndrome, especially since I had skipped my postdoc and gone straight from being a graduate student to a professor. And I think like virtually all new professors, you quickly realize that on a day-to-day level, many of the experiments that you want to do won't work. And really the best you can do is design your experiments so that even when they fail, they tell you something that still allows your research to move forward. But fortunately, I never ended up presenting those slides to my chair. And after a number of setbacks, second and early in my third year, things started turning around. We started to have a lot of success experimentally. We started to publish some papers that I was really excited about and got a number of grants by that time. And things started to look like they might develop in a positive, encouraging way to me. And of course, I continued to be very fortunate to recruit amazing students. And that's really been one of the best aspects of this job is getting to work with these really incredibly talented and dedicated groups of students and postdocs and eventually collaborators that have made it such a pleasure to come to work every day. And in a sense, such an honor to be able to work with students that are always in their intellectual prime. I mean, one of the downsides of being a professor is you only get the opportunity to work with a student or a postdoc for a handful of years before they leave. But the silver lining of that downside, and I think in this case, the silver lining is easily worth the price of the cloud, is that your students stay the same average age. They are always at their intellectual and creative prime, even as I get older and more senile. So being able to work with incredibly gifted coworkers, students, postdocs, collaborators who seem always to be in the prime of their creativity and their intellectual talents, and frankly, their physical stamina needed to be an effective full-time researcher. It's really something that I never take for granted, and that has kept me going for most of my career.
Definitely. I think that energy, that optimism can help bolster you during tough times. So I have to ask, David, when you were making this, we'll call it the failure PowerPoint, did you have a backup plan in mind? Like, what would you do if you had to leave?
I didn't really, and frankly, I wasn't thinking at the time about what was the alternative career route, in part because I had always wanted to and still wanted to be an academic scientist at that time. It was more this concern of how could I possibly let down this venerable university, this group of colleagues who were not only my very supportive colleagues now, but who had literally taught me from when I was a freshman in college. How could I best explain to them why I thought I wasn't going to make it and come up with a plan that would transition me out of this job in a way that minimized damage to the department and the university? It maybe just goes to show you how easy it is for us to create demons built on our insecurities, how easy and widespread imposter syndrome is. I've conveyed this story to students who have come up to me, postdocs who have come up to me and confessed their own forms of imposter syndrome, something very easy, very common to fall victim to in an environment where there are so many high-performing, very smart people that you're surrounded by. But I think overcoming our insecurities is such an important part of being able to do our best work. And it's something I really work on to maximize the culture of my lab, is to identify what are each student's insecurities and how can I best address them, and through examples in a public group setting and through private conversations with those students.
Definitely. I think this overcoming self-doubt is a huge hurdle for a lot of people in science, and I'm so glad you shared this story. But we don't just want to focus on the tough times, David. I'd love to share one of your success stories as well. So do you have a favorite success, whether it was a big win or maybe a small but particularly meaningful one that you'd like to tell us more about?
Maybe I'll tell the story about how we ended up developing prime editing, because it is largely a story about an amazing former postdoc in the lab, Andrew Anzalone. So in 2018, Andrew Anzalone, then a graduate student at Columbia in Virginia Cornish's lab, applied to my lab to be a postdoc. And I could tell from his application that he was a promising candidate. So I invited him to visit the lab and give a seminar on his work, standard postdoc interview. And during my one-on-one conversation with Andrew just before his seminar, so literally 20 minutes, 30 minutes before his talk, I asked him, well, what would you work on if you were a postdoc in our lab? This is a very standard boilerplate question. And most of the time, the candidates give general impressions. They might say, well, I'm particularly interested in your gene editing work, in your base editing, or they might say, I'm interested in protein evolution, or they might say, I'm interested in DNA encoded libraries, or occasionally they might talk about a specific idea that they have that they want to work on. And to be fair and realistic, most of the specific ideas at that early stage in the interview process aren't particularly compelling, either because I don't think they're feasible or because even if they work, I don't think they'd be that important. But Andrew's answer to this question was remarkable. He said, well, I have kind of the kernel of an idea for how we could copy new DNA sequence information encoding a desired edit, a change we wish to make, from a guide RNA directly into the genome. And it is inspired, he said, by your base editing work. But our base editors don't copy DNA sequence from a guide RNA into the genome. Instead, they perform a chemical reaction on an individual DNA base. But Andrew pointed out that what base editing had taught him is that the future of gene editing, in his opinion, was not cutting DNA to disrupt genes, but was doing what base editing did, namely converting one DNA sequence to another sequence so that you could correct mutations rather than simply disrupt target genes. And so I said, okay, well, that sounds like a very well chosen important problem. How do you propose to do it? And he sketched out sort of the basic underpinnings of what would eventually become prime editing. And there were a lot of uncertainties. There were a lot of holes in the idea. And I encouraged him, well, this is a little unusual, but would you mind presenting the skeleton of your idea, at least at the end of your interview seminar that's about to start? So he stepped into our kitchenette and he made a couple slides and he presented them. And I think the lab and I were both skeptical because the idea was so ambitious and required so many things that we didn't know would happen to go right. what Andrew and I would eventually call the 4 small miracles that had to happen in order for this project to work. And we, I think, all recognize that if it did work, it would perhaps be the most versatile way to do gene editing, because it would represent a true search and replace mechanism to change DNA sequence. You could replace virtually any sequence with virtually any other sequence using this approach. So I made Andrew the offer. He started working on this project, which we now call prime editing. And he showed a level of experimental skill and scientific insight that has never been exceeded in my lab's 24-year history. He made virtually no mistakes. That doesn't mean all of the experiments were a success, but he designed every experiment so beautifully that even the ones that did not work revealed why they didn't work and informed what the next experiment should be. And as a result of making virtually no wrong turns, Andrew completed this project and the 4 small miracles in an amount of time that I never thought would be even remotely possible for such an ambitious project. So it was 1 year and 8 days after he set foot in the lab for the first time that we submitted the manuscript to Nature that would eventually be the prime editing paper, published in October 2019. And that's still a sort of stunning level of continuous, sustained success for a year on a project that was incredibly difficult because, again, these 4 major problems that we had to either engineer a solution around or just hope that the cell behaved the way that we thought it might, but we really didn't know for sure. That's what led ultimately to the development of prime editing, which is now a very widely used method for editing genomes because of its unprecedented and still, I think, unmatched versatility in that you can make virtually any kind of change up to several hundred base pairs, either replacing letters with different letters, deleting letters very precisely, or inserting letters that are missing. And as a result, prime editing is now used not only as a research tool in hundreds or thousands of labs around the world, but is also the basis of a number of programs headed to the clinic, hopefully next year, that will use prime editing to correct directly the misspellings in our DNA that cause genetic diseases.
Well, David, this is an amazing success. And I love that you know the count to the day of the project timeline. I think that really speaks to just how remarkably quickly all of this really came together. And I know science can be all-consuming, particularly when you're working on one of these projects, one of these molecular mysteries, as you described it earlier, that you're really excited about. But we always encourage our listeners to step back, to kind of think more broadly, to read for fun. So we love getting book recommendations from everybody we have on the show. So David, do you have a favorite book that you can share with us that you've really enjoyed?
It's not a science book, but a book that I've enjoyed and reread parts of frequently for both entertainment and for life perspective is Trevor Noah's Born a Crime. I like books that bring me out of the academic science lab of what I think the public would stereotypically call the ivory tower and remind me of how important it is to connect with the world, either directly or indirectly, because so much of the world is very different than what any one person experiences every day. And so that's a book that comes to mind.
Well, excellent recommendation. We will add this to our website for our listeners to find there. And I think one of the other aspects of science that we haven't really touched on much yet today is travel. I think there are so many opportunities to see the world, to meet with amazing scientists all over the world. So do you have a favorite place, David, that your science has taken you?
I've been fortunate to have traveled to hundreds of places over the past 24 years, thanks to my scientific career. That has dovetailed very nicely with my hobby as a photographer. And in some cases, I confess that I've timed seminars, visits to specific places to coincide with times where I thought the scenery around, or in some cases, specific locations I wished to visit would be especially worthy of capturing in a photograph, like timing the visit to the Salk Institute so that my seminar would occur the day after the spring equinox so that the sun would set right over the sacred river that is the architectural feature that runs through the Salk Institute, or visiting Oregon Health Science University at the very peak of autumnal fall foliage season so that I could visit the incredible Japanese botanic garden in Portland and photograph some of the incredible, effectively bonsai trees, although they're not potted, so they're not technically bonsai, but they're these small, beautifully manicured Japanese trees in this garden that are just national treasures as far as I'm concerned. So I've been very fortunate to visit places around the world to be able to see the beauty and in some cases capture some of that beauty through photography as a result of science. So it's certainly a great career to pursue if you wish to travel, because there will be times in your career where you have far more invitations to travel than you could physically accept. And it's a wonderful opportunity to have.
Absolutely. I think it's wonderful that you're able to really tap into that passion for photography and science. And perhaps that in and of itself goes against some of the stereotypes that people may have of scientists. They don't necessarily portray scientists often in the media as these creative people who have a variety of interests. I think there are still some stereotypes out there that really show scientists in this negative light of being these solitary creatures working in a white lab coat by themselves in the lab all the time. And we love trying to break this stereotype by just highlighting some of the different aspects of scientists, their personalities, the quirky traditions that you have in the lab, and just some fun memories that you've shared. So do you have a story that you can tell us that really shows this human side of science that people may not always get to see?
Well, many stories, in terms of pursuing other passions, there have been so many, some of which have involved some level of self-deprecating, humiliating experiences. One that comes to mind, actually last night, I was doing some machining of a table that I'm making, and I needed to do a little bit of welding to complete part of this project. And that simply harkened back to how I learned to weld, which was during the pandemic. Like many people, I think we were all being driven a little stir crazy by not being able to go to our usual workplaces, to our usual social gatherings. I think there's a special kind of pressure and angst that experimental scientists that I certainly felt. I love either doing things with my hands or at least living vicariously through my students doing things with their hands to see how manipulating the world the way we want can result in interesting scientific discoveries. So I decided during the pandemic I was going to teach myself how to weld. So I bought a welding machine powered by electricity, put it in my basement, watched a couple of YouTube videos to try to learn how to weld. And I'm sure at this point, you and your listeners are thinking, what could possibly go wrong? Right. And of course, the first thing I did buy a welding helmet, so at least I didn't blind myself. But the first thing I did was light my clothes on fire multiple times. I have a couple long sleeve t-shirts, which have these wonderful holes in them where the fire had burned in some cases before I realized that I was on fire, since when you weld, it's a pretty intense, all-encompassing thing. It's very loud and a lot going on. But the reason I was interested in learning to weld was because I had this vision, which actually came from a dream that I had after, probably inspired by watching the Avatar movie, of these floating islands with beautiful vegetation. I'm sure anybody who's seen the Avatar movies can remember those scenes. And I decided I was going to build that and plant my next bonsai tree on a floating island. So, of course, the major technical hurdle is, well, how are you going to make an island float? So I could do the island. I molded the islands out of epoxy putty and paint and varnish and used inverted thinking stones, suiseki, to form some of the base of the islands. But how to get them to float? And what I decided to do was to hang a chain from the island downwards. So it looked like a sort of steampunk, vintage, mystical iron chain that was just dangling off of these islands. But the reality was that the chain links were welded together so that they were very stiff. But I would weld them in a catenary arc, in the arc shape naturally formed by a string that is allowed to dangle and come to rest away from its starting point. Forms a shape called a catenary arc. And so I became obsessed with welding a chain so that it would look like it was gracefully falling at an angle from these islands, but it would actually be stiff and strong enough that it would support the entire weight of these islands, which it turns out, because they're made of stone and solid epoxy putty, weigh a lot. So the result was I made 3 of these floating islands with chains that are formerly bonsai pots. The bottom of the welded chain ends in a slab of transparent epoxy putty that's on a very heavy base of petrified wood. The pots themselves then have, carefully drilled and hidden holes so that they have proper drainage, which bonsai needs. And then they end in beautiful bonsai trees at the top. And you can go on my Twitter account and see pictures and videos of some of these floating bonsai islands, which I continue to have to this day, several years later. I actually just repotted 2 of them. And it's an example of just an interesting art piece, really, that came out of a vision and a passion for trying to teach myself something in a somewhat awkward and humbling way, namely how to weld without injuring myself.
Oh, that is so cool. And I love that while lots of people were out there making bread, you were learning how to weld, and perhaps a more ambitious hobby during this pandemic. I think that is phenomenal.
It's a great hobby. It's surprisingly meditative. Just please be careful if anybody's thinking of taking up welding, learning this. It's actually pretty easy to do, but please don't light yourself on fire.
Yeah, maybe keep the extinguisher handy.
Yeah, wear clothes that cannot be set on fire.
Oh, that is phenomenal. And I know you are doing amazing things in the lab. We talked about some of them today, but I think oftentimes you're held back by different barriers, whether that's funding, staff, technology, or just feasibility, as we mentioned. So if we took away these barriers, David, and you sort of had carte blanche here and you could do whatever you'd like, what is the one research question that you would want to answer most?
I would want to develop the full suite of technologies and applications of those technologies of taking control of our genomes so that we have some say in our genetic destinies, in the health conditions, in the quality of life factors that result from misspellings in our DNA that we have no control over. And in some cases that offer the possibility of reducing our risk of disease. And I think achieving that goal will require continuing to develop not just new technologies for editing genes, but also technologies for delivering those gene editing agents into the right types of tissues in people, and the regulatory and ethical framework that's needed to maximize the access of all segments of society for these technologies, which I appreciate will take time, and to ensure that their application is as safe and efficacious as possible.
I think this would be absolutely transformative, and we appreciate you dreaming big with us on the show. And I'd love to end, if we can, with some advice. So, David, do you have a particular piece of advice that you received at some point in your career, or an inspirational message that you would like to end our conversation with today?
Find out what stimulates your curiosity more than anything else in the world, and then devote your life to satisfying that curiosity, scratching that itch. Because if you've really found something that truly captures your curiosity, work won't feel like work. Again, it will feel like the thing you would rather be doing than anything else in this world. And then the other piece of advice, which is a direct quote I received from another former mentor and now dear colleague, although never formally my mentor, but I effectively think of him as one in the past because he's been such an important teacher to me, both in the classroom and when I was a young professor, and that's Professor Stuart Schreiber. One of his mantras is stay within yourself. That is, we can spend a lot of effort trying to be who we think other people want us to be, or be who we think we should be, but maybe deep down inside we know we aren't. And you just achieve, I think, a much more peaceful life and a much more productive life if you stay within yourself, if you know who you are and stay true to that. And then finally, maybe the third answer to your question is a piece of advice I give maybe more frequently than any other to my own trainees, my own students and postdocs, which is don't worry about the things that you genuinely have no control over. Now, that shouldn't be an excuse to not try to effect change when you are able to, but there's plenty in this world and in the scientific enterprise that we actually have no control over. And even though it's a very human instinct to worry about everything that might impact our life, you can greatly simplify where you should devote your effort each day, where you should devote your time each day, if you live by this golden rule of don't worry about things you truly cannot control.
Well, David, these are 3 phenomenal pieces of advice. Thank you so much for sharing them with our listeners today. And if they want to learn more about you, more about your amazing research, perhaps see some of these cool art projects that you've worked on, what is the best way for them to do all of this?
So our lab's website is LUGroup, LIUGROUP.us, LUGroup.us. And my personal Twitter account is @DavidRLiu, DavidRLiu. And our lab's Twitter account is @LUGroup, LIUGROUP. So those are pretty much the only 3 forms of outward-facing media that we try to keep updated. But on our website, you can download all of our publications. Every paper that I've published in the past, we post free of charge to our website. And that's served as a great resource, not just for the public, but for ourselves as well. I probably use our website to pull papers every day. And we also make freely available for nonprofit use all of the DNA constructs we've created, the gene editing agents, the various proteins we've evolved, many, many other projects. We deposit them typically to Addgene, the sort of nonprofit library of DNA blueprints. And those DNA blueprints for our base editors and prime editors, for example, have been sent more than 15,000 times to, I think the last tally is more than 5,000 labs around the world. So we're really happy that the science that we've done has become widely used by other labs. And we're excited to see this field continue to develop in ways that will provide benefit to as many segments of society as possible.
Excellent. Well, listeners, definitely check out these resources, get connected on Twitter. And David, thank you so much for your time today. It's been a pleasure.
Thank you so much for having me. And I'm grateful to you and your listeners for their time. Thank you.
We appreciate you joining us, listeners. Wonderful to have you here as well. Thank you so much. And we hope to see you again for our next episode of People Behind the Science.